Complement system. This session will give you a very basic overview on one of the most active components of the immune system. So let us look at the learning outcome of this session. Complement system straddles innate and adaptive system as a major humoral effector system. So, along with antibodies, even the complement system eliminate the foreign particulates. There are several complement components that get activated to give rise to an immune response. The word complement was coined as these components complement the antibodies in eliminating the foreign particulates. On activation, the complement components interact in an organized and regulated cascade mechanism to lead to what is called as a membrane attack complex. The complement system can cause lysis of cells or microbes, opsonization, pro-inflammatory trigger, and immune clearance. Now, when you look at complement system, it was discovered by scientist Jules Baudet, who won a Nobel Prize in 1919 for his work on antibodies and complement system. As he was investigating bacteriolysis by immune serum, he found two factors that were involved. One was a relatively heat stable component, that is the antibodies, and the second was a heat labile complement system. So when he heated the serum, he found that the level by with which the serum eliminated foreign particulates decreased. So the antibodies remained, but on heating, the complement system actually got denatured. And so the immune response was lesser than when you have a serum that is not heated. So he termed these as alexins. Now, the relatively heat-stable antibodies, they generally cause what is called as agglutination, and these antibodies could also, sensitize the microbes to action of alexins. So basically, the antibodies bind to the foreign particulates, and this antibody antigen complex is actually recognized by the alexins. So they will bind to these antibodies, or they will be attracted towards these antibodies that have been bound to the antigens, and they would generally cause lysis of the cell to which the antibody is bound. Now, what actually devised a very simple test for the lytic activity, because the easily detected lysis of antibody coated red blood cells was possible, and this is what he called as hemolysis. So, to find out the activity of the complement system, he carried out what is called as hemolysis. Now, the complement system basically has more than 30 soluble and cell associated proteins, and all of these are called as complement, and they're denoted by C. Mainly, all of them are denoted by C. So, you have C1, C2, C3, and likewise. Now, they are definitely contributing to innate immunity, but they also contribute to acquired immunity. And most of these complement proteins in the blood are produced by liver, or they are present on the membranes of cells. Now, several components. So how does it contribute to acquired immunity or adaptive immunity? Several complement components actually can bind to receptors that are present on B cells. The B cells then get activated. They differentiate into plasma cells and memory cells, and that is how adaptive immunity is elicited by the complement system. The complement components are in the body present in the form of zymogens. That is, they are present as an inactive form, or they are in the inactive state until induced. Now, most components are broken down. Once they get activated, they are broken down into two subunits on activation. Say, for example, let's consider C3. So C3 on activation can get converted to C3A and C3B. So both C3A and C3B would have some role to play in the immune response, and majorly they are pro-inflammatory. There are three pathways by which the complement system function. One is the alternate pathway, the classical pathway, and the lectin pathway. Now, the alternate pathway is also responsible for homeostasis, while classical pathway and lectin pathway, they're all responsible for eliminating the for particulate matter and also apoptotic cells. So, as you can see over here, there is the C3 is the main component of the alternate pathway, and through a cascade of events, it is able to form a complex on the surface of the host cell, thereby making the host cell, either get lysed or get eliminated. Similarly, you would have in the classical pathway C1 as the major initiating complement. With lectin pathway, there is what is called as lectins, which can bind to components present on the cell, and thereby you would have eventually the immune cells clearing away the cell that has been bound to by the complement. So all three pathways lead to the same terminal mechanism. They all three pathways are active as a means of immune surveillance. That means they can recognize foreign particulate matters, and all three pathways are regulated to keep activation limited in physiological conditions. When it is a pathological condition, the regulation is different. When it is a physiological condition, the regulation is different. Now, the alternate pathway or the classical pathway and the lectin pathways, they are able to recognize molecular patterns on the pathogens, and it is not just one complement but several complements are able to do so. For example, C3 is definitely able to bind to molecular patterns on the pathogens. Now, what are the effector functions therefore? For example, membrane attack complex that is formed due to the assembly of several components on the complement components on the surface of the microbe leads to what is called as the membrane attack complex. Then you have, of course, what is inflammation. Then you have opsonization. You have interaction with the adaptive immunity. All these are effector functions. So you can see how the antibodies that are generated, along with that, when you have complement, then they contribute to the innate, and when B cells get activated, they contribute to the acquired immunity. So the ultimate goal, whether it is innate or whether it is acquired immunity, is the elimination of the pathogen. Now, therefore, let us just summarize what are the main functions of the complement system. The complement, on binding to the surface of the target cell, are able to form, after a cascade of events, something that is called as membrane attack complex, and by forming the membrane attack complex, it is able to lyse the target cell. Then it carries out a phenomenon which is called as opsonization. So when you have the complement binding to the bacteria, the bacteria, or if the component binds to a target cell, then that target cell or bacteria becomes more palatable for phagocytes. When you say palatable, means it becomes more tasty. So as an opsonin, so opsonin is when you have the complement bind to the bacteria. As a opsonin, it is tastier for the phagocytes. So it is able to recruit more phagocytes to enable engulfing and destroying the bacteria. Of course, it has a very important role to play in activation of the inflammatory response. So one can see that when the leukocytes are through extravasation enter the tissue space, then you can have the complement binding to these leukocytes and also to mast cells or basophils, and they degranulate them to release the mediators, primary mediators and the secondary mediators that can lead to inflammation. Also, It can lead to what is called as clearance of immune complexes. So if you already have antigen antibody complex present, then the complement can go and bind to these immune complexes, and because of its binding to the immune complexes, they are able to recruit phagocytes, neutrophils or macrophages, and these neutrophils and macrophages engulf the immune complexes and clear it. So this is the fourth function of the complement system. Therefore, as you can see, lysis, opsonization, inflammatory response, and clearance of immune complexes is just pointing to one thing, and that is elimination of the foreign particulate matter. So let us conclude. The complement system is active in a healthy person just for immune surveillance. That is, they keep going around looking for foreign particulate matters, and they maintain homeostasis. The system is regulated to maintain limited activation during physiological condition. On activation, depending on its context, the complement cascade can be of three pathways: classical, lectin, and alternate pathways. These cascades contribute to the innate system by pro-inflammatory and antimicrobicidal activity, and the acquired system through antibodies and effector cells activation. The effector functions of the complement include lysis, opsonization, phagocytosis, inflammation, and immune clearance. The complement system is appreciated to be a functional bridge between innate and adaptive immunity, enabling an integrated host defense to challenges posed by pathogens. Thank you.